Good afternoon. I'm Gary Morishima. I'm one of the co-chairs of the Oil Technical Committee. Want to welcome you to the 20th PSC Seminar on Environmental Change. There's really a lot more going on in this world than CO2 accumulations, storms, droughts, floods, and wildfires. Today we've got a couple very distinguished speakers talking about plastic pollution and tire contaminants. Susanne Brander from Oregon State and Tanya Brown from Simon Fraser University. Suzanne's going to talk about the secret ingredient, plastic, how microplastics and microfibers are infiltrating and impacting marine and freshwater food webs along the west coast of North America. Susanne received a PhD in toxicology and pharmacology from the University of California, Davis, and she's an assistant professor in the College of Agricultural Sciences at Oregon State University. She was also on the faculty at the University of North Carolina, Wilmington, from 2013 to 2017. Research is funded by NOAA, Department of Ecology, EPA, National Science Foundation, Oregon Sea Grant, and California state agencies. Her current focus is on the effects of stressors such as emerging pollutants, microplastics, and climate change on aquatic ecosystems. Susanne I'm going to talk a little bit about plastic pollution. Those of you who aren't really familiar with it, plastic is ubiquitous, it's everywhere. There are roughly 400 million metric tons of plastics that are produced each year. That production is scheduled to reach about 1,100 metric tons by 2050. And each year, roughly 9 million metric tons of plastic is finds itself in the ocean. And by 2050, it's projected to reach between 850 and 950 metric tons accumulation. There was an article that was published a while ago that managed to get a great deal of the press that 850 to 950 metric tons of plastic is roughly equivalent to the weight of all the fish that are believed to be in the ocean. Tanya Brown from Simon Fraser is going to talk about the tire chemical 6PPD-Quinone, which underscores the risks of non-point source pollution in Pacific salmon habitat. Tanya is an assistant professor at the Department of Biological Sciences at Simon Fraser University. She holds an adjunct professor position in the School of Environment at the University of Windsor, Ontario. Prior to her current position, she was a research scientist at Fisheries and Oceans Canada with the Ocean Sciences Division, where she led DFO's whale contaminant program in support of the Government of Canada's whale initiative. She has 20 years experience in the study of the ecology and ecotoxicology of marine mammals and fish. Her research is focused largely on understanding the influence of habitat use and feeding ecology on contaminant exposure, accumulation, and health effects in a changing marine environment. What is 6-PPD-Q? Well, this is a chemical that's found in the tires, toxic to coal, both adult juvenile and also juvenile Chinook. It enters our ecosystem primarily through rain and stormwater, finding its way into the waterways, causing some substantial loss of fish, not only salmon, but other species as well. So with that, I'm going to turn it over. And let's start.
Okay, thank you so much for the introduction. Thank you for having me to speak today. It's great to be in Vancouver, to come up here often enough from Oregon. Like was mentioned today, I'm going to be talking about plastics and fibers and basically how they're infiltrating marine food webs. And I'll focus on the west coast of North America today for good reason, but of course this is a global issue. I'll first start out talking about sources and exposure, talk a little bit about work that we do in the lab where we focus on how early life stages of organisms are responding to microplastic and microfiber exposure, and then talk a little bit about what we're going to do to move forward, some research that we're doing in our group in collaboration with Portland State University, as well as future directions globally. There's a UN treaty being negotiated right now on plastic pollution that I'll mention. So, as was alluded to in the introductions, microplastics are basically everywhere we look. These are plastic particles that, by definition, are less than 5 millimeters in size, but really our experience research-wise is that they are often far, far smaller than that. On average, what we're pulling out of a fish gut or a bivalve or sediment sample is on the order of 200 to 500 microns. So a bit smaller than 5 millimeters. And this is kind of a general listing of the different sources. Sorry, the font is a little bit small on this slide, but you can see that packaging is by far the biggest source of plastic waste, followed by textiles, which is sometimes surprising to people. But it turns out that if you look at where, on average, ocean microplastics are originating from, synthetic textiles are found to be about 35% of those, and that aligns with our findings in samples that I'll discuss a little bit later. And then car tires and city dust come in second and third. And so today I'll focus on microfibers and tire particles for the work that we're doing in the lab on exposures and responses. And of course, it isn't just about the ocean. Research is now expanding to include land-based sources, terrestrial sources, and we know, for example, that the agriculture industry alone uses over 12 million tons of plastic for plant and animal production. And that's gone up since then. And then another thing I'd like to bring up is that plastic production is really inherently linked to climate change, because often those two challenges are looked at separately, because it's made from fossil fuels. And then of course, as we're moving on in time, we're going to see that plastic share of global oil consumption is going to increase considerably. And that's in part because a newer strategy for the fossil fuel industry is to shift to plastics away from producing fuels, since more electric vehicles are now on the road and expected, that industry is expected to expand. So that's something that's happening globally with a variety of different fossil fuel companies. And a lot of this is happening, at a size scale that we don't necessarily think about or necessarily aware of. So I like to emphasize this because this is truly a micro-sized problem. So like I said, microplastics by definition are less than 5 millimeters in size, but this is a video of an organism we worked with a few years ago and published on. It's tintinid cilia, and it's at the base of many aquatic and marine food webs. Single-celled organism fed upon by larval fish, copepods, other zooplankton. And we found that in the lab, it readily takes up 10 to 20 micron sized particles, microplastic particles, and then those are transferred to predators. And literature more widely shows that larger predators tend to accumulate more microplastics from their prey than they do directly from the water. So what do we know so far? And really at this point, we have about a decade or more of data, at least on the marine and freshwater side, where really the problem was initially discovered. So most of the funding has been directed at those environmental matrices. Again, it's expanding to terrestrial sources now that we know that's probably even a bigger issue. But what we know so far is that the size and shape of particles are incredibly important. If you use the same polymer type and you do an experiment with a sphere, compare that to the same material that's made into a fiber, you're usually going to find that the fiber is more toxic. We don't exactly know why that is, but it may be because it's more difficult to eliminate. It may be because it tends to get caught up in the gut lining of the organism where it might be easier to translocate. Particles generally cause oxidative stress that can cause changes in behavior, and food dilution is a big issue. And that means what it sounds like it means. Basically, if you have a foreign particle stuck in your gut, and you can imagine, especially if you're the size of, say, a copepod, that that's going to take up a considerable amount of space, and you're going to get the signal that you're full. And so you're not going to go after food. You might not be able to absorb the nutrients from your food as effectively. And it does still remain true that many studies use purchased particles that are often spherical that really aren't environmentally relevant. So what our group aims to do is to conduct testing with particles that are made in-house. So we either cryomill particles from actual plastic products, so straws, cups, tires, which I'll talk about, and then we conduct high throughput testing with these particles. And this is done in my lab at the Hatfield Marine Science Center, as well as on OSU main campus in Corvallis in Dr. Stacy Parker's lab, who's a close collaborator of mine. But the high throughput testing, which is shown here, those tiny little dots swimming around in those wells are larval fish that are about 3 millimeters long. They're just a couple of days old. We've adapted this from testing with soluble chemicals that's been done for years to be able to get through larger numbers of particle combinations and concentrations to really get at what those responses are, at least over the short term, in these early life stages of organisms that are highly sensitive and also easy to work with because they don't take up, lots of space. You can do testing in well plates or small beakers. So a little bit about the plastic production that we do on site, and a lot of groups are doing this now, creating their own particles on site using cryomilling, which is basically grinding particles from products using liquid nitrogen and pressure. We also make microfibers on site, either using a microtome or cutting very carefully with a razor blade. You can see a polyester fiber at the bottom there, and those are created from natural materials to be as close as possible to what you would find in the environment. And this is all done with early life stages of fish. I work with a model fish called the England silverside, which is used nationwide. It's an EPA model species, and then a shrimp called the mysin shrimp, which is found off the coast of Oregon, where related species are, and they're consumed by salmonids and other important species. So we're finding that textiles are really quite a challenge, and this is an agreement with literature that's been published widely. This is from a study that we published in 2023. What we were noticing in our environmental samples that we were collecting is that we were getting a really diverse mix. We weren't just seeing polyester, we weren't just seeing plastic fibers. We were seeing a lot of cellulosic fibers, probably from people's laundry, right? Your blue jeans, your t-shirts are often a combination of polyester and cotton. So not surprising that we're finding that cotton in the environment, especially because even cotton is heavily chemically treated often. And so it's treated to be durable, which means it's going to last in the environment. What we found in this study was that cotton was comparable to polyester and polypropylene in terms of food dilution in mysid shrimp. And so if you fed mysid shrimp any of those fiber types over a week-long period, you saw that their growth was significantly reduced. That's concerning because it means that as a prey item, it might not be as valuable, right? Smaller, might have lower caloric value. That's something we're starting to look into. So a lot of the fibers are glued too, probably from blue jeans. Not to blame anyone who's wearing blue jeans today. But of course, this is, what we're also looking at is working with this across particle types. So it isn't just the fibers, is it other particle types that are causing this issue? And does it matter in terms of life stage? And it looks like these organisms are most sensitive in their first few days of life, when they're smallest, when there's potentially more of a long-term implication for effects on growth. But we even see this after a 3 or 4 week exposure. We see that microfibers, especially new microfibers, that probably still retain some of that chemical signature, even though we rinse and try to clean those fibers before we use them, We think that combination of chemical treatment and that shape of that particle really drives this effect on growth. And we saw that microfibers were the only particle type of this suite that significantly reduced growth in older fish. So we've also done some work on tire wear. And these again were controlled short-term exposures using the same early life stages of the same species of fish. And we actually were able to get a representative mixture of tire particles from the US Tire Manufacturers Association. They sent out particles to a number of researchers that are focused on toxicological responses like our group, or groups that are focused more on the chemistry, which Dr. Brown is going to talk about in a bit. We were focused on particle effects here. And so what we did was a dilution series, and we looked at concentrations increasing from 10 particles per mil all the way up to 1,000 or 10,000 particles per mil, as well as a leachate that was created by rinsing those particles and capturing what had rinsed off. And what we found is that the particles caused a decrease in growth, and that was consistent across most of the concentrations, with the exception of the lowest one. So in agreement with other research, other particle types. But we also saw effects on behavior. Why is this relevant? Well, behavior is important, especially in larval fish, because it's going to help them avoid predation. It's going to help them settle in an appropriate habitat. It's going to help them find food. And what we find is that leachate at a relatively low concentration, so nanograms per liter, had an impact on behavior. It affected mobility. It affected swimming distance in some cases. It also caused something called freezing behavior, where the fish just kind of stay motionless for a period of time. And all of these are indications of increased anxiety or neurotoxicity, which we'll talk about in the context of salmon salmonids too. So to get to the other sort of portion of our work in the lab, we do a lot of environmental occurrence work on a variety of samples. And this is quite an investment of time. It involves all of the steps shown on the slide here. So you get your sample, isolate and dissect. I have a student working on lamprey right now, for example. So watching her take the gut out of a lamprey is pretty fascinating. It just kind of keeps going. But anyway, so you dissect your tissues, you have to digest them using things like potassium hydroxide, which break down the biological material, but not the particles themselves. Then there's the inspection stage, so lots of staring down a stereo microscope, and we do material verification using Fourier transform infrared spectroscopy. Long story short, it's a lot of looking at filters like this after you've digested a sample. This is an example where this sample was actually spiked with different materials to get an idea of what our spiked recovery rate was, which is just as important for microplastics as it is for other types of analytical chemistry. You can see here there are some tire particles. This is a sphere that was spiked. This is a biological material. It's a piece of shell. So it's a lot of trying to differentiate between what's natural, what isn't, and then randomly selecting different particles to get an idea of what the composition of your sample is as a whole. So a few results from this work. We've worked on a number of different coastal species over the past 5 or so years. I started at OSU in 2017. So we did a study that was published last year on Oregon rockfish. These were black rockfish that were sampled from areas near Oregon marine reserves. So there's a program through the Oregon Department of Fish and Wildlife that was kind enough to donate samples to us that go out and do regular sampling outside of those reserves. And of course, reserves don't protect any fish from plastic particles. Not surprising. It's not as if there are walls and a roof over the reserve to keep them out. But what we found, not surprisingly, is that there were a lot of microfibers. So our microfiber occurrence was in agreement with larger global analyses that have been done on that particle type. And we found more fibers in the larval fish. So these are larval fish. These are near adults over here. than we did in the adults, but more larger particles in the adults. So probably reflects dietary differences, as well as differences in prey size. Okay. We also recently completed an analysis of metophids. A lot of these metophids were acquired from sampling that had been done by NOAA over the years. Rick Roger at Hatfield had, as many samples as we could possibly want, right? So student Olivia finished looking at these, and she was examining a mixture of metophids that have been caught from different stations off the coast of Washington and Oregon. Found that about 34% of metophids contained particles. And of course, again, the majority were fibers. What's interesting, and is in agreement with what we see in terms of fiber color and fiber characteristics, is that the majority of them were blue fibers that were cellulosic, so probably from blue jeans. And it turns out that blue is the most popular color for clothing worldwide. So all of these things. We've also had a great opportunity to collaborate with, through Lori Whiteham on these, the international year of the Salmon cruises. And again, this involved looking at microplastics in metophids as well as squid. We wanted to see if there was a difference between the levels found in these different species since they're feeding at different trophic levels. And have a question as to whether there's evidence of bioaccumulation or even biomagnification. And not surprisingly, we found microplastics. They're everywhere we look, right? So that's not news. But what was interesting was that the number of microplastics per metophid was 1.6 versus 3.9 particles per average in squid. We don't know for sure if this reflects bioaccumulation, biomagnification. That is the paradigm's a little bit less clear with microplastics than it is with, say, soluble lipophilic chemicals like DDT or other legacy pollutants. But there is an indication that there's this species difference and that squid may be consuming more microparticles on average. And just to kind of put that in perspective, for a Pacific albacore that's eating an average number of metophids for that species, you're looking at about 300 million particles per year, potentially, exposure to one Pacific albacore. I don't have the calculation for salmonids, but salmonids in the North Pacific also prey upon metophids and squid, so they may be in a similar situation depending on the volume they're ingesting. And then to kind of cap this off, talking about coastal food webs, another study we had an opportunity to be a part of was looking at how Pacific gray whales might be accumulating and be exposed to microparticles. And so we collaborated with the Gen Lab led by Dr. Lee Torres at Hatfield Marine Science Center and collected 3 different species of zooplankton that Pacific gray whales commonly feed on. Collected these off the coast using light traps and then brought them back to my lab and spent a long time splitting them up by species, doing their digestions and isolations and all those things. And of course, in the end, we found mostly fibers, right? Some other particles, green as fragments, blue as fibers. And then again, found that most of these were anthropogenic in nature, but not necessarily plastic. They were a mix of lots of different materials. Right? And again, these were coming from the inside of the zooplankton. And we were able to, thankfully, because of some great research done by graduate students, Hildebrand, previous to this, we were able to scale this out to figure out what a whale's daily exposure might be. And it's a little bit alarming if you think about it from the perspective of either a lactating or a pregnant gray whale. We know that these whales at those life stages ingest millions of mysid shrimp on a daily basis. Even if each of those mysid shrimp only contains on average half of a particle or half to 3/4 of a particle, you're seeing that there's the possibility for these whales to be ingesting over 20 million fibers a day. What does that mean for the whales? We don't know yet, but research on mammals suggests responses like oxidative stress, changes to the microbiome, things that are subtle on the surface, but over the long term might be really concerning in a longer-lived species. right? And so we're building on this by looking at responses in wild Oregon episodes. So sampling those, bringing them back into the lab and doing feeding exposures like we had been with model organisms that I spoke about earlier. This is an image of H. sculpta, which is a native Oregon mysid. It's also found on the coast of Washington and I believe Canada as well. And you can see that it's taken up a polyester fiber. So the student is going to be looking at things like caloric value and impacts on nutrient uptake and how that would scale up in terms of what a whale's needs are in collaboration, again, with the Gem Lab at Hatfield. But really, just to sum this all up, this is a really complex and challenging problem because we're not just looking at one plastic type or even one material type. We're looking, I feel like I'm basically going out into the field, bringing back fish and then finding people's laundry in these fish, right? That seems to be about where this has ended up. And it's not just that, it's also other particle types that we don't necessarily think about, like the lining of coffee cups, for example. We're finding fragments that even resemble the film that lines your typical disposable coffee cup. And a lot of this we're doing via FTIR, so this is an example of a spectra. And those databases that microplastics and microparticle scientists use are still really being built out. And so this is an open source database where any reading that you do and enter into this program, it's put online into a database so anyone globally can access that information. And so it's gradually building out and allowing us to have more accurate identification of these particles. But it's still a real challenge and we're still sort of building on that information and growing that field. So in summary, over the short term, growth and behavior are impacted in early life stages. And that's shown in work that we've done from my lab and from other labs as well. And food dilution doesn't seem to be dependent on polymer type. It's more so dependent on size and shape. For example, I didn't present these data, but we even see that bioplastics like PLA induce the same behavioral and food dilution responses. Over the longer time periods, fibers have the biggest impact on growth and other parameters. And it seems that low levels of ingestion in zooplankton can really translate into sizable microparticle exposure, depending on the predator's dietary needs. Fibers can translocate. And I didn't present these data either, but looking into other sample types from the land, some of these things are coming from sources that we don't necessarily even think about, like biosolids from our wastewater treatment plants that receive all of these fibers from our laundry and then are respread onto ag fields and other locations to use as fertilizer once they're sterilized. So there's a whole Pandora's box that's sort of left to explore in terms of where these particles are coming from. So what can we do? I'd like to end sort of on a semi-positive note here, because that's a lot to digest. Oregon Sea Grant has just funded Portland State and Oregon State to focus on a study where we're going to be installing filters and washing machines in several coastal towns to see if it reduces over time the number of particles that are loading into those coastal ecosystems. And so that's a study that has just started because each load of laundry could release as many as 9 million microfibers. We're also going to be looking at stormwater management and installing bioretention cells in those stormwater outfalls from those same towns to get an idea of whether that can also reduce both microfiber and microplastic fragment loading into those coastal ecosystems. So stay tuned, we'll see what we find. I'm hopeful. But long story short, globally we have a big problem. Only 9% of plastic waste is recycled, most of it is mismanaged, and the flavor of mismanagement varies depending on where in the world you are. In the US, we're pretty good at management, but we're really bad at recycling, it turns out. We only recycle 4% of our plastics, even though we think this is a long-resolved issue. It's not. Even though we design our trash cans to be mostly recycling, we're not mostly recycling. And the UN has taken this on. In 2022, they agreed on a mandate to a legally binding treaty addressing the full life cycle of plastics. They've been meeting regularly over the past couple of years and will meet again this coming April. And I'm part of a group called the Scientists Coalition that is aiming to provide the delegates to this UN negotiation with accurate science-based information. And so trying to convey things like thinking about this across the entire life cycle, from sourcing and polymer production all the way down to waste management, removal, and remediation. So some positive things are in the future there, should that treaty go forward, which we think it will. Wanted to end with a giant thank you to everyone who was part of the different studies I mentioned, and also a thank you to the Pacific Northwest Consortium on Plastics, which is our research group at OSU and Western Washington University that has been doing a lot of this more targeted lab exposure work. And you're welcome to join the consortium if you're interested in learning more about what we do and get our presentations. Thank you so much again, and I'm happy to take questions if there's time.
Hi, and good afternoon, everyone. Thank you so much for inviting me and for the introduction today. As it was mentioned, I'm going to be speaking to you today about the tire-associated chemical 6PPD-Quinone, and discussing some of our work that we've been doing, but also just in general, what is 6PPD-Quinone. With respect to Pacific salmon, I think we can all acknowledge that Pacific salmon are important to First Nations, recreational and commercial fisheries, as well as our resident killer whales, both the southern resident killer whales and the northern resident killer whales. There are a number of populations that are both endangered or threatened. And with respect to contaminants, there's a significant gap in knowledge with respect to the concentrations that we find of contaminants, as well as the effects in adult and juvenile salmon, originating Canadian waters specifically, but also even south of the border. And this infiltrates both within their freshwater and marine habitats in terms of our lack of understanding and knowledge. And with respect to contaminants, there's over 30,000 chemicals that are identified that are with respect to Canada's marketplace that are produced in high volume. And that's if you make it even less specific, there's over 100,000 different chemicals that are used. And contaminants have been identified as one of the greatest threats to our natural ecosystems, and specifically to juvenile salmon health and survival. So 6PPD-Quinone, which is the chemical I'm speaking to you today on, is what has been identified as a contaminant of concern with respect to, especially with respect to coho salmon, but also with respect to other salmon species. And other species are being studied throughout not only Canada, but down in the US, but globally right now. The trouble with salmon in particular is that they have a complex life history and habitat needs, and that's not something I need to go into much detail with respect to with you folks, but just with the context of contaminants, they're exposed to different chemicals throughout their journey. So if you're thinking of a juvenile salmonid leaving their freshwater environment, you may be more concerned with, say, wastewater treatment effluent and more contaminants that are posing a risk from a local perspective, rather than when they get to the marine environment, their exposure is both to a local and global perspective of contaminants. So just in the context of mercury, for mercury to make its way over from Asia, it takes about 7 to 8 days for it to make it to our waters over on this coast. So just to put that in the context of the local versus global perspective of what contaminants are being exposed to throughout their journey. For 6PPD-Quinone, there's been 20 years, over 20 years of research with respect to not 6PPD-Quinone, but certainly with respect to urban runoff mortality syndrome. And that was first noted in Washington State with the toxic injury and death of up to 90% of adult coho salmon that were returning to the urban and semi-urban waterways around the Puget Sound area. So this was first reported in the 1990s in urban streams around Puget Sound, and then later linked to increased stormwater runoff and tire-associated contaminants. And this was through decades of research, of intense research down in Washington State and other areas. And then, of course, in 2020, when the Tian et al. Paper came out, it was linked to the 6-PPD-Quinone, which is the byproduct of the parent compound 6-PPD. So this is the 6-PPD-Quinone chemical here. And as I mentioned, 6-PPD is the parent compound. So what you get from that, you get 6-PPD-Quinone that's released from tire rubber, which is converted by the ozone to the analog 6-PPD-Quinone. So 6-PPD, the parent compound, is far less toxic than the breakdown product 6-PPD-Quinone. So with respect to 6-PPD, the parent compound, it is added as a tire additive to tires, and it's present in tires at up to 2% of the weight of a tire. So if we're thinking about the number of chemicals that are used in tires, you're looking at around 400 different types of chemicals that are used in tires. So, you know, we're talking to you about, what I'm talking to you about today is one chemical. So just putting that in the perspective of 30,000 in high volume, but also with respect to the 400 chemicals that are actually used in tires. And then if you're thinking about the loss of tire particles, you're looking at about 20% of their weight being lost in about the first 5 years of driving. And there's been some new research showing that with electric vehicles coming on stream, even more so, they're likely to use even more tire chemicals, up to 30%. I think I read more recently up to 50% loss compared to fuel-powered vehicles. So this has generated a number of road runoff renewed concerns about fish habitat, potential effects to not only coho, but other Pacific salmon, as well as other species in general. And then it brings us back to a lot of the other contaminants that are present in road runoff, like tire and paint particles, road salt, hydrocarbons, metals, detergents, etc. So 6-PPD-Quinone is just one of many of the cocktail of chemicals that are being received into our salmon-bearing waterways. So this is a study that we conducted over the last couple of years, where we were looking at the effects of 6-PPD-Quinone on juvenile salmon. And we were using really a multi-omics approach. So what I mean by that is we're looking at the kind of upregulation and downregulation of your genes endpoints in the salmon, as well as linking that to how their metabolites or hormones are responding to this chemical. And then we're linking that to more tissue level responses, where we're looking at the histology of different organs in the tissue of question. So we're looking at 4 different species, where we looked at coho, chinook, chum, and steelhead over a 24-hour exposure period, so an acute exposure. And then of course, we were measuring with this particular exposure, we were looking at the LC50. So that's the lethal concentration, where you'd expect 50% of the population to be dying. So this is one of my PhD students that's co-supervised with my colleague Vicki Marlett at SFU. This is Bonnie Lowe. Some of her research was published last year, where we found that juvenile coho were 3 orders of magnitude more sensitive to 6-PPD-Quinone than juvenile chinook. So what you're seeing here, the 2 graphs are showing you on the y-axis is the percent mortality, and then on the x-axis is the concentration of 6-PPD-Quinone. So that lethal concentration, when we're expecting 50% of the population to be dying, we're seeing that the lethal concentration is 41 nanograms per liter in the water, whereas with Chinook, we're seeing greater than. We weren't able to even calculate the LC50 because of the solubility of 6-PPD-Quinone, as well as the fact that we weren't seeing the lethal die-off in the Chinook salmon, but we were anticipating greater than 67 1,000 nanograms per liter. So considerable difference across species. And if this is just a table to kind of summarize some of the literature that's been published to date and what we know from other salmonid species. And what we're finding is that coho remain the most sensitive species to 6PPD-Quinone, and that's followed by brook trout, rainbow trout, and then Chinook salmon. So and I just want to mention that some of this work is agreed upon with what was documented earlier with response to lab controlled studies with specific road runoff exposure. So they're looking at the range and sensitivity across those specific salmon species. And that was a paper done by French et al. Down in Washington State. So some of the symptoms of acute 6PPD-Quinone toxicity that's been documented in the literature. Just to give you some indications, and maybe you've seen some videos and so on at other meetings or online, but generally you get this, there's cardiorespiratory, neurological impairment going on, and where you get surface swimming and loss of equilibrium. So it's a very distinct type of swimming behavior that you see within the first 5 to 6 hours of exposure. Some of the physiological symptoms that have been documented is increased, really what they're finding is a blood-brain barrier disruption, and then the increased oxygen consumption rate, which goes hand in hand with some of the cardiorespiratory behavioral symptoms that I mentioned above. And then some of the work that we were working on right now in a paper that Bonnie Lowe is working up is that we've been seeing structural changes to the gill. So this is one example on the right, in the image on the right here, where we're seeing basically this individual would not be surviving much longer if it was you were to monitor it post for another 1 to 2 hours. So stepping back away from some of our exposure and effects work that we've been doing in the lab, we've also been carrying out a number of field studies for the last three years, where we've been going out and sampling 6PPD-Quinone and other road runoff associated contaminants in our salmon-bearing creeks. So we've looked at over 80 different salmon-bearing creeks. And what we typically classify as our rain event that we go out all year round is a greater than 48-hour dry period. We aim to have at least 5 millimeters, a minimum of 5 millimeter forecasted rain event. And then we typically sample before, during, and after the rain event to start to get an idea of what are the baseline concentrations of 6PPD-Quinone in salmon-bearing waterways. What are the concentrations reaching it during a rain event, and then how quickly are they coming down after the rain event ceases within a 24-hour period. So this is one example of one of our creeks here in Metro Vancouver, Burn Creek, just giving you an idea of what the first flush would look like in one of our salmon-bearing waterways. So very dark, dirty water. And then whenever we go out, we're doing a number of different measurements. So we look at how quickly is the water flowing. We also look at we take a number of physical and chemical measurements. We're looking at dissolved oxygen, conductivity, and so on. And then we also take water samples not only for 6PPD-Quinone, but we also want to look at what is that relationship with the parent compound. What does it look like with other tire-associated chemicals like diphenylamine, which is another industrial use chemical, not only in tires, but in other industrial uses. And then also with respect to metals, to get that idea, just to better understand what's the relationship that we're seeing with this chemical with other tire-associated and in general road runoff-associated contaminants. So this graph is showing you our average concentrations that we detected in a few sites, both within Metro Vancouver on the left-hand side. So this is 6PPD-Quinone in nanograms per liter on the y-axis. And then in Victoria, on Vancouver Island, and in Sydney on Vancouver Island, to give you an idea of what we're seeing before, during, and after. So the orange bars are what we would be the average concentration during a rain event. And then after would be within 24 hours after the rain has stopped. So on average, what we're seeing is that the levels of 6PPD-Quinone are approaching or exceeding the effects concentrations in coho during our rain events. And that's irrespective of season and so on. So continuously, certainly within our Metro Vancouver sites, all of our Metro Vancouver sites, we would expect to see lethality for coho salmon. And then I guess I also forgot to mention, just in terms of that increase, you're seeing up to about a 90% increase from that before, which is our kind of our background concentrations of 6PPD-Quinone, so what you would find anytime you're going out in the waterway and sampling, to that during rain event, and then it comes back down. These images are just giving you an idea between, so on the left-hand graph here, you've got the full change. So just try to standardize between like the background concentrations of 6PPD-Quinone and the concentrations we collect during a rain event. And just to look at the number of dry days, because we are seeing sort of in certain years a greater number of dry days, whereas certainly within Vancouver and along the coast, we're seeing those dry days, and then we're getting rain events and so on, especially this year in particular. So there is a relationship, and this is just a subset of data from one of our sites for serpentine, but I'm working on the paper right now, and we're continuously seeing this across a number of sites. And then on the right-hand side, the plot here, this is just with respect to flow. So how fast is the water flowing, and what is the relationship showing with respect to 6PPD-Quinone? So as one would expect, as the water's coming up, you're getting an increase in the 6PPD-Quinone concentrations. And then this graph is just giving an indication of just the relationship that we're seeing between 6PPD-Quinone and then another tire-associated chemical, the diphenylamine that I mentioned to you. So diphenylamine, we do know that it is toxic to rainbow trout, but very limited information is out there with respect to its toxicity. It is used in tires, as I mentioned, but also as well as a number of industrial chemical applications. So I think this relationship of between these two compounds just, raises new questions about tire chemistry, as well as just our understanding and the importance of looking at not only 6PPD-Quinone, but other chemicals and the relationship it may have with posing a problem or risk to Pacific salmon. So I just want to put this into perspective, going back to some of our lab exposure work with Chinook salmon, and posing that question, are Chinook salmon at risk in our urban waterways? And I think, if we look at the lethal concentration at 25%, so we actually have a value of that, and that's 43,000 nanograms per liter. Again, considerably higher than what our lethal concentration is to coho, but also considerably higher to what the concentrations are that we're actually measuring in the field. So what's been reported in our work so far is we're looking at about 0.1 to around 12,000 nanograms per liter during rain events. In the 12,000 would be around Metro Vancouver, where we're seeing the highest concentrations of 6PPD-Quinone. And then in salmon-bearing waterways, similar concentrations, and that was from the Tanya Brown paper. Stormwater runoff, and if you're looking at pure road runoff, you're looking at concentrations in the order of around 2,500 or 3,000 nanograms per liter. So it's not to say that Chinook salmon may not be at risk, but from a lethal perspective, it doesn't appear they're at risk. But certainly if you're thinking about the sublethal effects, I think, this is a growing area of research that is underway, and I think we need to just look at that in a little more detail. So just to bring us to a close here, I think we can all acknowledge with respect to our British Columbia here, salmon 6PPD-Quinone has been impacting, has very likely been impacting the health and survival of coho salmon in our urban and semi-urban areas, and this has been going on for decades and decades without us really knowing it. Again, Chinook, chum, steelhead, and so on are far less sensitive than coho, and that continues to be supported through the literature. We currently, with all our 80 sites, and we're continuing to collect a number of other sites, but then do some modeling work to do a hotspot analysis across coastal BC to show where 6PPD-Quinone is posing the greatest risks. And this will assist with prioritizing sites for initiatives to mitigate impacts to these sensitive species. And then to the effect that it's affecting salmon stock abundance, I think this is a larger question at hand, but it does obviously still remains unclear, but ultimately this data is informing mitigating measures. Some of our work that we've are building out in support of our monitoring work that we've been doing on the collections of the salmon bearing streams for water. We've got new sites around Metro Vancouver that we've added to, and we've split off into 2 teams with teams from my group and then teams with colleagues of mine that have involved within a project at UBC with Rachel Scholes and Tim Rogers. And we're doing a more comprehensive analysis where we're going to start to identify specific sites of green infrastructure. And this work is being, Tim's using some of this work to help indicate and inform municipalities on where we should start to put in some of our rain gardens or bioswales and so on. We've also been looking at measuring how effective retention ponds and rain gardens and bioswales are at reducing 6PPD-Quinone and just using different field sites. This is a site where there's a retention pond there in the Mackenzie interchange on Vancouver Island, where we've been sampling before and at the pipe and before and after as it enters into the Goldfoot Creek. And then options for mitigations, I've sort of touched upon some, but of course, rain gardens or bioretention cells. There's the recent paper done by Tim Rogers showed that it could reduce the loading of 6PPD-Quinone by more than 90%. And then just thinking about automotive engineering possibilities, there's the Tire Collective in Europe has piloted a prototype to collect tire particles from your tire when you're driving. And then of course, sustainable tire chemistry. So looking at replacement chemicals that could do a similar, prevent that, the wear down from tires, from the sunlight and so on, which is why we have 6PPD introduced into our tires. And then, of course, just bringing us back to ultimately the regulation of this toxic substance. In the case in Canada, that would be done under the Canadian Environmental Protection Act. So yeah, just want to acknowledge everybody that's contributed to all the collection, and I'm going to got a lot of my core group listed below. There's many people involved in the collection of all those field samples and then involved in even the lab studies. And then funding to support a lot of this work has mainly been through a number of different grants through Fisheries and Oceans Canada. With that, I'd like to thank you, and yeah, happy to take any questions.
Well, thank you. I think that very interesting and informative talks, and a bit scary. I think that it begins to open our eyes to understanding that we've been doing a lot to our environment over a lot of years. And in order to address and recover salmon, there are lots of different things that we've got to think about and figure out how we're going to deal with. Susanne, Tanya, thank you very much. And we're going to take a few questions here. Lori, where's Diego has some microphones. Those of you who have questions, please bring them forward.
Tanya, thank you very much for that presentation. I work quite a bit with Interior First Nations. I'm particularly interested around the 6PPD accumulation issues as it reflects to 3 major coho producing tributaries in the interior Fraser that are right beside major highways, namely the Coldwater River beside the Coquihalla, the Eagle River beside the Trans-Canada Highway between Revelstoke and Seminar, and also the Alberta system, which is in the upper North Moxon. Interior Fraser coho are living in what we refer to as a reverse hydrograph, and those fish are largely, the juveniles are largely living in off-channel groundwater fed ponds associated with those rivers, and those highways go straight through those floodplains. The groups that I'm working with would be very interested in working with you, if the group would be interested in studying the effects on those systems. We know that we have an issue with road runoff in the cold water because we run a resistivity counter to count fish coming into the cold water, and just the road salt spikes alone coming off the cold water on a rain event or a rain on snow event are enough to throw the resistivity counter off where we can't use it. So we'd be very interested in those ponds basically are acting like retention facilities.
Yeah.
So it has the potential to have a major problem. So we'd be very interested, if you were interested in working with us to look at some of those issues. Thank you.
Yeah, thank you. Yeah, we definitely, if you can reach out to me, I can, afterwards, I can provide you with my email and phone number, but yeah, we'd be happy to look at those and all of our work, for the most, we have a core team that goes out and does a lot of the sampling, but really, our program wouldn't be possible without all the partnerships that we have from NGOs, First Nations and so on, and stream keepers going out and sampling. So we're happy if you got, and a lot of the sites that we've identified are through people expressing concerns and that are on the ground observing and want their, specific areas looked at. So yeah, we'd love to do that.
Okay, my name is Leah Mellinger, the fish program for Port Campbell Small Drive, and I was wondering, I've heard a lot about 6PPD or QPPQ, and other research that I've done at UC Davis and working in partnerships. But one of the things I've not seen is, does it degrade in saltwater as far as oceans? Because a lot of the research has been done on accumulation and runoff in your river systems, but the river systems go into the bays, go into the sound. So I was curious on whether or not we have an idea of any accumulation, or does it just dissipate in ocean water?
Yeah, I don't know of any published papers yet that have recorded measurements in ocean water. I know that there was some research that was presented at a recent toxicology conference this past in 2023 that was reporting concentrations measured in Chinook salmon and things like that. So in adult Chinook salmon, and then I may have the wrong species, but adult salmon. And then I think they also looked at even in killer whale, blubber, and things like that. So that would be colleagues at NOAA that were measuring 6PPD-Quinone. So there is work starting to be underway in tissues, we are doing it in estuarine water, but I'm just thinking about ocean water. We get the dilution effect with respect to 6PPD-Quinone, but I don't know of any studies that reported it in the ocean.
I'm more interested in the fact that we have, or the tribe has a cocoa net pen, and considering how sensitive they are in compared to chum and chinook and steelhead, if there's a pollution, is it enough, or could that be contributing to some mortality, international needs, and that sort of thing? So I'm just curious, especially in the mid canal, whether or not that particular toxicant is playing a role.
Like with respect to even, I'm thinking about like the areas that we're seeing the highest concentrations there, these are low flowing waterways, creeks, streams, and so on. And you're getting that pulse, right? And so that pulse is coming down within a 24-hour period. So you'd have to have kind of an ongoing pulse to be getting elevated concentrations that would likely result in lethal toxicity.
Thank you.
I have a question. Is it known why cocoa is particularly sensitive to this component compared to others among its, 3 orders of magnitude?
The short answer is no. But there is a lot of research underway. So there's lots of colleagues, my group's been looking at that. There's colleagues across Canada, a few other colleagues that have been looking into that in terms of just comparing different salmon species. Rainbow trout is another species that has a little bit more sensitivity relative to other salmon species. So that's been used as another species to kind of look at, what might be helping to explain what's going on in coho too. And then, of course, people down in the US and Washington State especially. But no, I don't think we don't have that answer yet. But it's very perplexing. And when you're thinking about the regulation of chemicals, sometimes we regulate chemicals in the context of just their physical chemical properties, and we look at it that way. But we also, then we look at it in the context of using a model species, whether it's Daphne or rainbow trout. But then when you have a chemical like 6PPD-Quinone, well, then if you're just protecting, say, Daphne and rainbow trout, this chemical wouldn't have been even on the radar. And it wasn't on the radar because the parent compound was passed through our legislation regulation. So, you get it can be the case where we even with DDT as well, those breakdown products are more toxic than the parent compound, so.
Thank you both for these terribly depressing presentations. My question to the room is, how do we even have salmon left? And it blows me away that these species are surviving. So back to Dr. Brander's microplastics presentation, I'm really disturbed by that projected increase in plastics over projected for 2050. That is really it's very intriguing. So, are there any signs that you may not be working on this, but anybody working on, are there adaptive changes being seen in organisms or evolutionary change where smart organisms are not eating plastics, and you're starting to see shifts, they're just dying off, right? Is there anything that's showing up that give me a little bit of ray of hope here?
I'll try my best. I'm not always the best at giving rays of hope out, but I'll see what I can do. I would say the short answer is no, the fish probably aren't going to adapt, and in part because especially the predatory species are acquiring these from their prey, so it's not as if they are intentionally going after a microfiber or a fragment, or they're inhaling it through their gills as they're uptaking water to excrete oxygen. So I would say no, but the encouraging thing is that globally we are already working towards a solution with these global treaty negotiations. And from my perspective, as someone who's been a toxicologist, I started my first faculty job in 2013, and I've worked with other classes of chemicals, pesticides, endocrine disrupting chemicals, different pharmaceuticals, plasticizers. And there was really no movement forward whatsoever on most of those chemicals, especially the pesticides, whereas the plastics field has already moved to we know this is bad enough that we need to do something about it, and we're already acting at a global level there's been suggestions that we even need to cap virgin plastic production by 2040 and only reuse what's already out there. And so different strategies for recycling, establishing circular economy. So that I see as the ray of hope that globally scientists and policymakers are already coming together to try to find a solution. It's going to be tricky and messy, but it's happening. So, hopefully that's enough hope. Try it.
Hi, Suzanne, this is for yourself as well.
Okay.
Question for you. I'm curious about the translocation to the, you indicated the fillets.
Yeah.
So is there a particular type of microplastic, particular shape or size that's translocating? And is there evidence that maybe there are fragments of larger particles? So I'm just curious about, is there any signs that these microplastics are being processed in a way that would allow for them to be broken down?
Sure, that's a really good question. So a group of us, aquatic toxicologists and chemists, did a meta-analysis that we published a couple of years ago, looking just at aquatic organisms from studies that did document translocation. And what we ended up coming up with was that there's a cutoff of about 60 to 80 microns for aquatic organisms in terms of the ability of particles to translocate. It does probably depend a bit on shape as well, and the microfibers might be particularly good at translocating because they are so narrow along that one dimension. And so they can be as narrow as 10 to 12 microns on average. And so you can almost think of it, a colleague of mine described it as like a spaghetti noodle going through a pore that's large enough to fit that through, and that's probably why we're seeing them in fillets. And initially the response to them being documented in fillets was, well, you must have background contamination. This must be shedding from your clothing, not coming from the environment. And then there have been a number of studies published showing the same thing. So we're certain that that's probably what's happening. It's just difficult to visualize, to document that. For mammals, as far as what is known up to date, particles need to be a bit smaller, so below 10 microns and maybe even as small as 5 microns in order to translocate. But it's happening across all living organisms at this point. Yeah, it's really a size issue. And in terms of breakdown, that can be happening with it's happening with particles down to the nano size. So I don't know if you saw the study about particles found in bottled water that came out a couple of weeks ago. They were able to, using a new technology, they were able to measure the concentration of smaller particles in the water. Then normally we have a cutoff of about 1 micron because spectroscopy approaches like FTIR and Raman spectroscopy don't let you visualize anything below 1 micron in size, but they were able to use a new approach, which the name is escaping me at the moment, but they could look at nano-sized particles. And because they did that, they found much larger concentrations of particles, because as you get into the smaller size fractions, you're getting orders of magnitude more of those smaller sizes. So long story short, there's probably a lot of translocation going on. You know, what it's doing, we don't have a full understanding of yet, but it has to do with just the tendency of plastics to break down into smaller particles until they're, you know, we haven't looked at the, you know, the size below nano yet, right? I'm forgetting what that begins to be, picomolar, but you know, that might be the next frontier. So we should just use less plastic. Okay.
Mine is more of an observation. We're doing a lot of work down in Washington State, Northwest Indian American Tribes, and our Environmental Policy Committee. We've been tracking the tire dust and we've been tracking microplastics. We've been all these environmental outliers, including replacing our culverts so they're fully functioning culverts. We want a court case for that. Anyway, you know, we've been talking here at the Pacific Salmon Commission about, well, at least I believe we know that we can't harvest management our way out of our losing our salmon. And this is great presentation that says, here are the other issues that are affecting the survival of our salmon. environmental, and chief among that is climate issue, how that's changed. Our water is low, it's warm, we don't have the spring runoff for out migrant juveniles. When we get the spring runoff, now they're full of tire dust. And then there's a rise in the fall, one function helping our juveniles, the other one killing or impacting. In Washington State, we're plugged in, we're actively working through our congressional delegations, the House and the Senate, the appropriate committees, interfacing with tire communities. They're telling us they need something short of a decade to figure out what they can change or how they can change. We've got one or two of our tribal representatives on national chemical, I can't even remember the acronym, let alone say it what it is. They're meeting on all these toxic chemicals, you know. We're fully engaged, and how do we all work or influence this work that needs to be done? I think the end result of tire dust is if they change that changes that 6PPD-Q or Quinone question. I'm thinking that does, but it might be wrong. And then how do we align ourselves and so we're all working and get this stuff addressed in a more effective or efficient way? Because I think in the last decade or longer, we've been zeroed in on these kinds of issues. Stormwater runoff is a huge issue. Washington State can see and know that it's everywhere where salmon are. I just appreciate that we're having these kind of discussions because it isn't harvest management. You can't manage anything. If there's no more salmon, we don't exist as an organization. And quite frankly, us in Washington State, our treaty tribe, without salmon, we don't exist as people, our culture and our way of life. So what I'm wondering is how do we build momentum to keep these kinds of discussions to get results in these areas where our salmon can't survive. And I just wanted to thank you folks for all your hard work.
And back to the question on the translocation of the particles into tissues, is there work started now to look at the impact of that to the predators as well? So you're bringing those particles, you're finding in the tissues, what is the impact of those organisms? Thank you.
So we've been doing some work on this with the fish species, the model that we work with quite a bit in the lab because we've been able to sequence the genome, and so we can look at things like gene expression. And I don't have these data to show today, but what we are showing when we look at RNA-seq, which basically lets you get a snapshot of all of the genes that are being expressed at a particular moment in an organism, is that there are lots of different pathways being affected, everything from cardiovascular to neurotoxicity. I know that there have, not in our lab, but other studies have shown that the nano-sized particles can cross the blood-brain barrier. So that makes sense that we're seeing that. But really, using the RNA-seq approach, we were able to see responses that aren't always documented. You know, when you pick a couple of genes, you know, cherry pick to kind of look at, because most studies are showing, you know, reactive oxygen species production, which is going to be produced anytime the animal is under some kind of stress. But it seems that microplastics that are small enough to translocate are able to disrupt things like circulation. They're translating potentially into the brain, also seeing things like reproductive disruption, which you see with endocrine disruption and just endocrine disrupting chemicals. but these are particles that are probably causing that via a different mechanism, because they're not, binding to, the estrogen receptor, they're doing it in some other way. So yeah, I think the predators are being affected in a similar way to the organisms that are consuming those particles directly. It's just a matter of, as a toxicologist, you're always aiming to sort of understand what we call an adverse outcome pathway. And that for microplastics, it's so complicated because they don't act like a molecule, right? It's not just looking at a molecule that you can say, oh, that's going to fit into this receptor or this enzyme, right? And so you're kind of, it's really kind of blows the door open in terms of the possibilities. So that's why it's taking a long time to really understand what pathways are being affected. But yeah, that's the short answer.
Just one more question about the microplastics. There's been some research on basically bacterial degradation possibilities of microplastics. If there's a ray of sunshine, do you know, or have any deals? I know your background's toxicology, but what are your thoughts about that, those types of work?
Yeah, good question. We have a great microbiology department at OSU, and I had a chance to collaborate a little bit with them. PhD student went down to the Bahamas, so in Oregon winter, so we were all very jealous of him, and collected some plastic, typical environmental plastics, and was looking at the specific bacterial families that were growing on these plastics, and did find that there were several potential candidates that could be used, say, in a wastewater setting or waste management setting that would break down those plastics. I think that is one tool in our toolkit, in addition to just limiting. I mean, the first need is to reduce at the source. But if we are able to specifically select certain bacterial species that will break down the most commonly used plastics, I mean, that would definitely be a huge step forward. It's just a matter of most of the bacterial candidates he was finding broke plastics down relatively slowly. So, you put them on a little well plate and breaks it down over a couple of weeks, but just thinking about the volume that would need to be dealt with, it would need to be scaled up quite a bit. But there's work on that front that's encouraging, for sure. Yeah.
Excellent presentation by both of you. On the second presentation, I believe there was a statement made that electric cars are more damaging than gasoline cars. Is there a reason for that?
My understanding is it comes down to weight. So because electric cars weigh so much more, in part, you're wearing down your tires in a much quicker pace. So I think there may be some other variables in question, but that's one thing that can explain it.
Oh, me again. Sorry. Thank you. Quick. So I did pick up on your table. Did you have an LC50 for chum salmon?
I don't know. I don't.
There was one?
No, we have one. Well, we don't have, we are publishing one in the works, but it's gonna be a greater than value.
Greater than?
Then basically the solubility of the chemical itself.
Oh, okay. So it's similar to like.
It'll be similar, it'll be around Chinook, exactly, yeah. And same as far, my understanding, a colleague, I think has worked on sockeye, and sockeye is quite high as well. And there was a paper published by French et al., and I'm happy to share that with you. But that wasn't done with the chemical 6PPD-Quinone itself, but it was done stormwater runoff. So essentially, the results we're getting in the lab to 6PPD-Quinone are mimicking what has been found previously in terms of sensitivity.
Okay, so because those studies are on juvenile salmon, correct?
That's correct, yes.
And then we see a lot of the symptoms of the results in adult salmon.
Yeah, we see
So
it
you've got
in
these urban
both.
streams
Yeah.
with coho and chum. The coho are the ones that are having a lot of trouble with it, not the chum. So,
Yeah,
that ability to deal with this seems to persist.
yeah, from a legal perspective, but I think there's still many, many questions to be answered in terms of what are we seeing at the sublethal like,
Oh, for sure.
right? So we want to send our salmon out to sea in good shape so they can return and spawn and so on, right? So what's happening to them in our creeks before they go? So I think that's a important questions to look, that needs further research and understanding. So yeah, I think we've got a good understanding from the lethal concentrations and sensitivity range, but some lethal effects is still remains to see. Yeah, oh,
Can I ask a question?
yeah,
I was looking at the slide where you were describing the symptoms, that were demonstrated by the coho salmon, and the difference in sensitivity between the different salmonid species is just fascinating
Yeah,
to me
it's
from a
crazy.
tox perspective. And they increased hematocrit, ionic dysregulation, the cardiac function is declining, and it made me think of a slide that I showed to my students. I teach an environmental physiology of fishes class. I'm just wondering, and someone's probably looking at this, but is anyone looking at the ionocytes in the gills of the coho versus the other species and how they might differ in their sensitivity?
there may be, I'm not sure, to be honest, it's
Yeah.
quite possible that somebody's already looking at, but we have not looked at that yet. We've been really focusing on just trying to get some of our RNA-seq result, like the kind of molecular results and so on. And then I have another postdoc that just started with me that's going to be looking at doing co-specific exposures as well in salmon-bearing waterways to look at a number of different changes. So, but yeah, that's a good question. Yeah.
Cool, thanks.
All right, folks, we're at time. I want to give a big round of applause to our two speakers. Also, I'm struck by how new these disciplines are and how many data gaps and how much knowledge you've already acquired. So I'm excited to see where these disciplines will be in 10 years. So thank you very much.